to show them my note card. Okay, everyone wait. Okay. Human genetic modification. When you hear those words, you might have thought of the designer baby. You can personalize your designer baby with the color eyes that you want, the color hair that you want, and even the level of intelligence you want. This concept is highly controversial, but it isn't actually what human genetic modification is. So today I'm going to be telling you a little bit about it, just in case you're worried about custom-made children running around in the future. I've done a lot of research on this subject, and I do find it to be very interesting. So today we're going to be getting into the technology used in human genetic modification research, how that technology is used today, and finally some of the arguments against it. So, what exactly is human genetic modification? Well, according to Nancy King, gene editing is the technical process of deleting segments of DNA from an organism's genes and sometimes substituting new sequences of DNA in order to eliminate deleterious mutations. So the technology for this process was first introduced in the 90s as zinc finger nucleases, but it was actually highly inefficient and very difficult to accurately pinpoint genes because of how complex the system was. The CRISPR/Cas9 system has been developing over the course of the past 20 years, but it wasn't until 2013 that it was actually adopted for genome editing. So CRISPR actually made it far easier to target genes in DNA sequences. So scientists started using non-viable embryos and messing around with their genetic structure. This resulted in a lot of controversy, but also a lot of studies conducting research and speculation in scholarly reviews in journals. So the next thing we're going to be getting into is the use of human genetic modification today. I want to make one thing clear. No one in the US is picking genes out for their children for enhancement purposes. The National Institute of Health, the National Institute of Science, as well as Congress and the FDA have all come out and said, we are not endorsing this and this is not going to happen under our watch. So no reputable clinic is going to be able to make your kids' eyes blue or give them the ability to run like Usain Bolt. So what is it actually used for? Well, it's used for editing genes that are linked to blood disorders such as sickle cell disease and beta thalassemia. A lot of studies have been using non-viable embryos, but according to the MIT Technology Review, in 2017, Shukrat Mitalpov of Oregon Health and Sciences University was able to successfully extract genes from viable embryos that are responsible for life-threatening disorders. These embryos were not allowed to develop for more than a few days, so they never actually turned into fetuses. This was a major breakthrough for the biotechnology industry. But the commotion that this caused resulted in a lot of people commenting about how CRISPR should be used today. So the next thing that we're going to be getting into is some of the valid arguments against human genetic modification. So I've already gone over the concept of designer baby, which is one of the main arguments used against human genetic modification amongst the public. But the main argument coming from people who are actually involved in this research and industry is that one, it's very, very likely that you accidentally alter off-target genes. And two, that the embryos, we don't know how it's going to affect embryos in the future. Edward Lamphere, who has 30 years of experience in the biotechnology industry, says that genome editing in human embryos using current technology could have unpredictable effects on future generations. And in a study in 2019 by Wuxing Yu, he and his team found that while it is possible to significantly decrease your chances of altering said off-target genes, it's still possible. So this could have a lot of effects on not only the embryo, but also that embryo's future children, should that embryo even be born. Everyone in this industry is calling for open communication or open conversation and communication about their findings. So that includes everyone in the government, people who would be funding this kind of research, the people conducting the research, and anyone who this research is being conducted on. So hopefully today you guys learned a little bit about human genetic modification, namely the technology used in its research, how that technology is used today, and lastly, some of the very valid arguments questioning its ethicality and safety. This area of research has a lot of potential for good use, and it can be kind of scary to think about, but you don't have to worry about any custom-made children running around just yet. Does anyone have any questions, Dad?
Can you splice dog genes with human genes?
I don't think so, no. Okay, any other questions? Yes.
Where did you say Nancy King worked?
At Wake Forest University. Do you have a question?
So, I'm not super versed in this area, but if I was going to have a child and I just wanted to, and I knew that I had bad genes of something like like a disease or something, you're saying this could X out that disease?
Potentially in the future, yes, but it's not being used right now. It just research is being conducted. No babies have been born in the United States using this technology.
Any babies, any other countries using this technology?
China has claimed that they created twins using this, but there is nothing. When the U.S. asked them about it, they refused to comment.
Okay. Wow, this is so interesting.
All right, I think that's good. It's fun.